We've made it to Unit 3, Biodiversity. Hooray! Let's start with a deep time review. When did eukaryotes appear in the fossil record? About 2.5 billion years ago, as oxygen began accumulating in the atmosphere. We also need to review classification because these larger, more general groups evolved earlier, and more specific groups evolved more recently. In Unit 1, we focused more on the genus and species levels. Now we're going to look at these larger categories of domain, kingdom, and some phyla. Let's start with domains. There are only 3 domains for all life forms. One is bacteria, another is archaea, and the third is eukaryota. These domains are based on cell structure. Every life form on Earth has one of these 3 types of cells. We already learned that eukaryotic cells have a nuclear membrane, an added layer of protection for DNA. Karyo means nucleus. Eu means true. So eukaryotes have a true nucleus. Pro just means before. So prokaryotes evolved before the nucleus. For a long time, we thought there was just one type of prokaryotic cell, bacteria. But it turns out that there's 2, bacteria and archaea. Here's a very simple graphic comparison. There are slightly different proteins that stick outside the cell membrane. It's these dots on the outside of the cell. Bacteria have one type, and archaea have another. There are slightly different fats that make up a cell membrane. Bacteria have one type, archaea have another. And there are slightly different proteins that read the DNA to start the process of transcription. Here's the cool thing. If we look at a eukaryotic cell, it's a combination of the bacterial and archaeal traits. Eukaryotic cells evolved when a bacterial cell and an archaeal cell combined. Here's a very simple phylogenetic tree showing these domains of bacteria and archaea. The first life occurred around 4 billion years ago and split into these 2 prokaryotic groups. But about 2.5 billion years ago, they combined and evolved into a small population of eukaryotic cells. This event was an example of endosymbiosis. Endo means inside, as in inside the cell, and symbiosis, of course, means working together. It's thought that a large archaeal cell engulfed a smaller bacterial cell, and the 2 began working together, one inside the other. These eukaryotic cells then began getting food or energy in different ways. Plants experienced a second endosymbiosis event engulfing a photosynthetic bacterium that allowed the cell to make its own food. Animals take in food, ingesting it and digesting the food within their body or cell. And fungi grow through their food, digesting it outside the body and then absorbing the nutrients. These 3 types of energy acquisition can be categorized as autotrophic, which means the organism produces its own food, or heterotrophic, the organism takes its food from another life form. Plants are autotrophs. Animals are heterotrophs. What about fungi? Fungi are also heterotrophs. This shared characteristic with animals is important in identifying evolutionary relationships. But before we get to that, let's look at this last traditional kingdom, the protists, or single-celled eukaryotes. Back in the 1960s, protists were all lumped together. But over the past 2 to 3 decades, with new DNA comparisons and analyses, we've realized that they're not closely related at all. There are actually an estimated 14 kingdoms of protists. But don't worry, because I'm not going to make you learn 14 kingdoms of protists. But we do need to make some changes because of this. That change is a classification level between the domain and kingdom levels. Before we get to that additional classification level, let's look at this split between the heterotrophs and the autotrophs. In this unit, Unit 3, we'll cover the heterotrophic branch of eukaryotes, organisms that take food into their bodies to get carbon and energy. In the next unit, Unit 4, we'll look at the autotrophs, organisms that can take carbon into their bodies from the environment, from carbon dioxide, and make their own energy. And we'll also briefly touch on bacteria and archaea in Unit 4. Now for this classification level between the domains and kingdoms. These are the supergroups: Amoebozoa, Opisthokonta, Excavata, Archaeplastida, and SAR. This is an abbreviation for 3 groups that are still kind of being rearranged a bit as scientists learn more about them. These names may seem a bit overwhelming, but remember, we're spending 2 complete units on these supergroups. So before you get freaked out, remember, we're just going to be looking at these 2 supergroups for Unit 3, the heterotrophic ones. And then in Unit 4, we'll look at these 3 supergroups. So it's not that bad. If we start from what we know, you've probably learned that there's a kingdom of animals and a kingdom of fungi. That's in this Opisthokonta supergroup. You've probably also learned about the plant kingdom. Archaeplastida means ancient, archae, like archaeologist, and plastida, as in chloroplast. This group includes the plants as well as red and green algae. Animals, plants, and fungi are still kingdoms, but there are another 14 or so kingdoms that we just grouped into this protist group, otherwise known as the junk drawer of life. Why is learning about a kingdom called Protista a problem? Why am I making you learn these supergroups instead? As you can see, protist is not a clade. These organisms are all over the place, so there's no single evolutionary ancestor in common that includes all these descendants. So throwing protists into one group just pretty much says that we don't care about these things. But these life forms are just as important as any other, and they deserve to be studied and understood, even if it's just for our own personal gain. When we understand clades and evolutionary ancestry, that helps us make better predictions and decisions as well. For instance, some species within these different groups cause disease in humans. Which disease or parasite do you think would be most difficult to fight off? A bacterial infection? amoebic dysentery, cordyceps fungus, giardia, or malaria and apicomplexan. The fungus would be most difficult to develop some kind of medicine to help us fight off that organism. Bacteria is generally the easiest because their cells are so different from our cells. We can take an antibiotic that disrupts the physiological processes in a bacterium, but that do not affect eukaryotic cells. Fungal infections, though, would be the hardest because their cells are so similar to ours. And the amoebae would be the next most difficult because we're all in this one clade together. We've known about some of these clades for over 20 years, yet we keep teaching them the old way. At some point, we do have to change, and that point is now. If you go into medicine and end up working on malaria, just as an example, the malarial parasite is in genus Plasmodium. It's an apicomplexan. If we look at the taxonomic classification on Wikipedia, here is that SAR clade. It's in this alveolata group, and then in phylum Apicomplexa. If you want to fight this parasite, you need to understand its evolutionary history. It provides all sorts of information about the organism's niche, its physiological properties, cellular makeup, everything.